Okay, folks, this is it. Your last chapter of Human A&P. Not only that, but it's a pretty easy one because you've probably had a lot of this material in past courses, maybe as early as middle school, So some of this will be review, which is good because genetics can be a little bit confusing. So sometimes it's just good to reiterate some of these things because you'll use it time and time again in biology classes. But the important thing for today is to link heredity and, these genetic theories to how they, are carried out in reproduction in humans. And the big thing here is, we want to make sure that we are conferring good traits, that we are providing variability because we want to, keep being unique individuals, right? We don't want to be the same as our parents. We also want to make sure that we are not passing on dangerous diseases, which unfortunately is pretty common in humans and in all other animal species. You just have bad mutations that pop up, and unfortunately a lot of those diseases can be inherited because they are like a mutation within our genetics, within our DNA somewhere. So a couple terms that I'm sure you've had most of these before. Genetics is just the study of heredity. We have sex chromosomes, which are those X and the Y. And then we have autosomes, which is everything else, right? So sex chromosomes make us male and female, but they also have a lot of other genes on them. That's not like that's all they do. The autosomes are just every other chromosome, but the sex chromosome, the karyotype is this picture that you see on the slide. So it's just like a scientist arranges the chromosomes in this way so they just are all, matched up with their partner. So this is what that process looks like. You have to find all of the chromosomes, stain them, and then pair them up as best you can to try to align them. And this can be good for for various reasons, but the main thing is to see if you have any genetic disorders like Down syndrome that you could be testing in utero, like a fetus before a baby is even born, to kind of know some of the diseases that fetus could have. You can check for them this way. Okay, so we have sources of variation, and variation is really the building block of evolution. So, this is how we have grass and we have whales. Like, how do we have organisms on this earth that are so dramatically different? We have variation that occurred over billions of years. And so there's different ways that we can get that variation, and you just put it all together, which allows you to have even greater variation because these are all happening all the time whenever you have sexual reproduction. So segregation and independent assortment, these kind of go hand in hand because they happen when chromosomes line up as tetrads at the metaphase plate during meiosis. This does not occur in mitosis, only meiosis, because remember, mitosis is just you making more skin cells or any other cell in your body that are just like for regular, parts of your life. But meiosis is when you actually want to make a new organism, so you want to allow all this variation to occur. So you have these tetrads that line up and allow mom and dad's genetic material to mix and be passed on, differently to different offspring. So they're going to be distributed differently. The chromosomes themselves will be distributed differently. And then you can have independent assortment. Just if you think about it at the allelic level, you can have crossing over events that happen between the alleles as well on the different chromosomes, like they kind of swap some genetic material. But this allows us to have a lot of variability. Okay, so this is what that would look like. So each of these boxes is a different possible combination of mom and dad chromosomes. So, this is like, let's say this is, the result of this is going to be an egg cell. So, an individual woman got chromosomes from her mom and from her dad, and they line up at the metaphase plate during meiosis, and they can either all go into daughter cells, as they were inherited. So maybe all mom's chromosomes stay together and all of dad's chromosomes stay together, but that's definitely not always going to happen because they can be in any of these other combinations with just as great of chance or probability. So this just means independent assortment means that chromosomes from your mom and chromosomes from your dad are going to be put into new egg and sperm cells independently of one another. You're going to have a jumble of mom and dad's chromosomes that you are going to then pass on to your own offspring. So recombination is just taking that one step further. So those chromosomes are still lined up in their tetrads, but then they're going to exchange some genetic information. And this only happens in the homologous chromosomes that are like physically touching. So they're going to replicate their DNA, which is happening right here. So that's how you get the homologous chromosomes, because two chromosomes replicated their DNA and they make this X shape, and then they line up at the metaphase plate. And then the ones that are actually physically touching are going to exchange some genetic information, which means mom's chromosome now has little bits and pieces of dad's chromosome on it, and dad's has some of mom's genetic material on it. So you have this, further step of variability that can occur. And then random fertilization just means that all of the gametes that come from mom are just as likely to be passed on to an offspring as another egg from mom. And all of dad's sperm cells are just as likely to be passed on as another. So that is random fertilization. It's just like grabbing a random sperm out of a bag, grabbing a random egg out of the bag, and fertilization occurs. And you get siblings that look drastically different from each other because, any of those is just as likely as the other to occur. Now, when we talk about actually inheriting these traits from our parents, we have different types of inheritance, which I'm sure you've heard of. So dominant and recessive just means that if you have two alleles, and an allele is just some variation of a gene. So if you got a chromosome 17 from your mom and a chromosome 17 from your dad, and like right at the tip of my finger is where some gene is. If mom gives you, the trait for, if we talk about flowers, that's always easier. Like the mom gave red flower trait and the dad gave white flower trait. Those are alleles, individual variations of that gene. So the gene here would be color of your flower. Dominant and recessive would mean that one of those traits is going to cover up the other if they are both given to an offspring. So if we have red and white flowers, flower alleles, if you give both of those to an offspring, so they have both possible allele combinations, that flower is only going to be pink because the pink is going to cover up the white. So there are quite a few diseases that occur this way, which is good because if you are a heterozygote, as in you have the allele for one of these diseases, as long as you have the allele for like the normal, not having the disease, that will cover up the disease allele and you will be a normal, healthy human. A carrier means that you are a heterozygote, but you have the allele for the disease. So even though you don't have it, you can continue to pass it on to future generations. And then if you have a child, like you marry somebody who also is a heterozygote, you could potentially have a child that is homozygous recessive and will have the disease because they have two alleles for the disease. And this is a reason that consanguineous marriages are not legal in much of the United States, because if you have a certain trait that, runs in your family, if you marry a first cousin and have kids with that cousin, you're way more likely to pass on those harmful traits than if you're marrying somebody from another family. So that's why it's not just a stigma of being bad, it's also bad genetically speaking, because you can pass on a lot of traits that are not good, and you start building up all of these diseases in the same family, and you keep passing them on from to future generations if you have these consanguineous marriages continuing. So an example of diseases don't always have to be the recessive trait. unfortunately, in some cases, they're dominant. And an example of this is astigmatism, which is like, your eye is kind of football shaped or egg shaped versus perfectly round, which gives you blurry vision. That is a dominant trait. So this is more common in traits that, you notice that something's like not great, a physical characteristic like an astigmatism, but it's not something that is going to kill you. So they stay in the human population because those people still, have children and pass these traits on to future generations. But a lot of these disorders are going to be recessive because that allows them to stay in the population, because maybe somebody doesn't know they're a carrier and their spouse doesn't know they're a carrier, and suddenly they've both passed it on to their children and their children could have the disease. So heterozygotes, just remember, they can carry, but they're not going to have the trait if we're talking about a recessive disease. So consanguinity, this is kind of a look at what that looks like throughout the world. So it's very common in other places of the world, which, kind of contributes to why certain diseases are common in certain places of the world. In the United States, generally, it is not allowed, or at least very frowned upon, but there are still some states that allow first cousin marriages, and that's even more common throughout the rest of the world. But genetically speaking, not something that you want to do. We can also have incomplete dominance. So if we go back to our flowers, if you have a red and a white flower, incomplete dominance means that if a daughter flower gets the red and the white allele, that flower is going to be pink. So just somewhere in between. Sickle cell disease is like this. So if your dad has sickle cell disease, your mom doesn't, and you get an allele from both of them, you can be somewhere in between. So you can have some normal cells and some sickle cells. So this is not quite as common, but there are several other diseases that are inherited this way. So this is what that person's blood sample might look like. They have some healthy cells, but they also have sickle cells. And, again, this is something that's probably going to stay in the population because it makes you more likely to survive and pass these traits on to future generations. Okay, multiple allele inheritance. The easiest example of this is the ABO blood group. So this is just when you have more than two alleles. And not only is this multiple allele inheritance, but we also have a couple other inheritance types happening here. So A and B are both dominant, which means they're co-dominant, and both of those are dominant over O. So if we have two people, one of them has A blood, what is the person's with A blood genotype going to look like? Well, they can either have two A alleles, or they can have an A allele and an O allele, because the A allele is going to cover up the O allele. So it could look like this, two A's, or they could have an A and an O allele. So same thing with type B blood. What if you have two parents, one with AB blood and one with O? Pause the video here. On the next slide, I have those Punnett squares. You can write this out on your own and try to look and see if you can figure this one out on your own. I'll put some sort of Punnett square thing on the exam probably, so just kind of refresh this. It's easy once you do it a couple times, and all the other Punnett squares will come back pretty easily to you. And I'll go over them really quick here. So pause the video. Try to do this really quickly on your own, and then I will go through it here for you. Okay. So parents A, B, and O, can they have a child with type O blood? We're going to figure that out by just doing these examples. So AB blood, they have to have big A and a B, because those are the only two, that's the only genotype possible. The person with O blood, they can only have little I, little I. So what are the potential offspring from these parents, AB blood and O blood? They can only have children with type A blood or type B blood. So this is kind of one of those weird instances where none of your offspring are going to have the same blood type as the parents, none of the children. So there's a 50% chance you'll be type A and a 50% that you'll be type B. What about the second question? A person with type AB blood and a person with B blood, can they have children with type O blood? Well, we know that the person with type AB blood is going to be AB. The person with type B blood, the only way that they could have a child with type O blood is if both parents are giving the O allele. So the answer here is no, but let's just look at this really quick and see what that looks like. Even if the person with B blood donates an O allele, still none of those children can have type O blood because the O allele is recessive. And if it's going to be, presented in one of the children, they're always going to have type A or type B blood still, because that O allele always gets covered up because it's recessive. Alrighty, what about sex-linked inheritance? There's nothing weird about this other than the gene that you're talking about is on a sex chromosome, but it's inherited in the same way. The only difference is that guys only have one X chromosome and then they have a Y. Females have two X chromosomes, and the X chromosomes are way bigger than Y chromosomes. So you have a lot more genes that are going to be present on them. So a sex-linked trait is pretty likely going to be an X-linked trait, not a Y-linked trait, because it's just, the X chromosome is so much bigger. It has 2,500 genes, while the Y chromosome only has 78. So that's why most sex-linked disorders are going to be found on the X chromosome. Males are more likely to express this because they only have one X chromosome. So if the disease we're talking about, or just, generally speaking, a trait, it doesn't have to be a disease. If the trait we're talking about is a recessive one, if the guys have it on their X chromosome, they're always going to display that trait because they don't have another X chromosome with the dominant allele to cover it up. And then we have Y-linked chromosomes, which are even weirder because they can only be passed on from father to son. That Y chromosome, if your dad has a trait like hairy ears, he's going to pass it on to his sons only, because he has to give them the Y chromosome with that trait on it. If that son does not have any sons of his own, that trait stops there in that family. No future children can have it because it has to go directly from a father to a son. And you can see how different these sex chromosomes are, and it becomes very apparent why so many of them are actually found on the X chromosome and not the Y. Okay, so let's go through this quick example. We have a rancher. And ranchers, of course, like to breed their bulls with really good traits to their cows with really good traits to get even, potentially the best offspring they can. But this bull that this rancher has, for some reason, has this weird eye issue where there's no pigment in the iris. So the part that makes your eye a certain color, there's no pigment there. And that makes that bull more likely to have blindness or other, issues with the eye. It's going to be very sensitive to sunlight coming in. So the breeder wants to breed his bull still, because the bull has other good characteristics. So he wants to breed it with a cow that is not going to pass on this trait to any future offspring. So the rancher has two cows, and he's had the genotypes done on both of them. And we know that this trait that the bull has that leads to no pigment in the iris is a sex-linked recessive trait. And he has two of his cows genotyped. One of them is a big N, one of them is a big N, little n. So what is this going to look like if we cross either of these cows with the bull? What would their potential offspring look like? You can try this one on your own by pausing the video here and trying to go through it. So we want to know, between these two cows, what is the likelihood that we're going to pass on this no pigment in the iris trait to a future offspring? And the rancher's basically trying to decide which cow he should choose. Okay, so we know that we have the bull, and the bull has the little n, has that characteristic on his X chromosome, and then he has a Y chromosome. So the cow that he's going to choose should be the cow, of course, without this trait, right? Big N. Of course, we don't want to choose the cow that could also pass on this trait, because that's going to give us a greater chance of having an offspring with it. But what are the chances that any of these offspring, even when you cross it with a completely normal cow that's not even a carrier for the disease, you have a 0% chance of having any offspring with the disease in this case. You can have two females that could be carriers and give it to future offspring. So maybe you wouldn't want to breed any of the female offspring of this bull with any future generations. But the males are going to be normal because they will inherit the dominant allele from their mom, not the recessive X chromosome from their dad that has the trait on it. Okay, so all the offspring in this case are going to be normal. You could do the Punnett square on your own for if you bred the cow that has the little n, has the trait, and is a carrier for it. Obviously, you would have a greater likelihood of having offspring with it. The females will be carriers, so your recommendation to the rancher would probably be to choose this cow, but then don't let any female offspring of that pairing, that cow and bull, to have any offspring of their own in the future. You should eliminate that trait from the herd completely by doing that. Okay, and then polygene inheritance, this just means that you have multiple genes. So up to this point, even with the ABO blood type, we were just talking about the same gene and different variants of that gene, which is to say different alleles of that gene. But we have a lot of traits in humans. Anything that has like a ton of variability, like height, skin color, intelligence, hair color, is going to be inherited by multiple genes. And so skin color is inherited from 3 separate genes. All of them are a dominant recessive situation. So if you have A, big B, big C, that means you're going to have the darkest possible skin color. Little a, little b, little c means like the lightest possible skin color. So that's going to be at either extreme on this chart. But then, of course, we also have environmental factors that can affect these things. So it's not just like a simple outcome in a lot of this stuff, because you have multiple genes affecting traits and you have environmental factors that can come into play. So this is the reason why these diseases, autism, cancer, diabetes, cleft lip, and there are others, any of these conditions are really hard to treat and hard to, weed out of a population because you have so many genes that are influencing this trait in some way. Okay. Now we know, you've probably heard of the nature versus nurture debate, and we know now that it's not really a debate, it's just a combination of both of those things occurring. So you can have environmental factors that affect your actual genetics, like for the rest of your life. So they don't just, like a virus makes you sick temporarily, bacteria makes you sick, but any of these things like drugs and pathogens in the womb, in utero, can lead to lasting effects in the DNA. So it's why it's important to, monitor things while a pregnancy is occurring, but then after birth, things can affect your genetics as well. So nutrition and different hormones can affect gene expression, like what gets turned on and what gets turned off, like, the ability to produce certain proteins and enzymes. Couple other weird examples, non-traditional inheritance. Your book briefly talks about this. So genomic imprinting is when only your mom or your dad's allele is going to be expressed. So maybe you get one from each of them, an allele from each, but for some reason, only mom's or only dad's is going to be turned on. And that probably has to do with some other, factors in your DNA that cause certain traits to be turned on and off. Extranuclear inheritance is mitochondrial DNA. So not only do you have DNA in your nucleus, in your cells, you also have mitochondria that, probably evolved themselves from some sort of protein and just kind of like happened to get into animal cells a long time ago. And we just kept them there because they're good at making energy. They have their own DNA. not very much of it. They only have a few genes, but you can actually have diseases or traits that are tied to your mitochondria. And mostly those are going to be inherited from your mom, because when dad's sperm fuses with the egg and fertilization occurs, a lot of dad's other organelles and stuff are going to be lost. They're not going to be passed on and included in that future offspring. So Parkinson's is believed to be inherited this way. Obviously, this is going to be a more rare form of inheritance because you just don't have that much mitochondrial DNA in the first place. Okay, pedigrees. This is a way that you can recognize certain traits in your familial history and how they might be passed on to future generations. So a pedigree is simply tracing genetic traits in a generation or several generations in a family. So this is really beneficial when you are talking about certain disorders and you're trying to figure out the likelihood of passing that disorder on to future offspring within your family. Achondroplasia is a disease where you have short stature because you didn't have proper long bone growth. And this is actually a dominant trait. So if you have the allele for achondroplasia, you will have the disease. Even if you're just a carrier, you can't be a carrier in this situation. To not have the disease, you have to be a homozygous recessive individual. So little a. Anybody with a big A is going to have achondroplasia. In other words, they are going to be affected, which makes it more likely to be passed on to future generations as well. Hunter syndrome. This is going to be an X-linked trait, so sex-linked and X-linked specifically. So we're going to go through an example here, and we want to see the likelihood of Hunter syndrome being passed on to children. So if you were a genetic counselor and you were trying to counsel parents that came to you and said, we have a history of this disease in our family, and we want to know if we should even have kids or not, or should we opt for adoption, or an egg donor or a sperm donor if we know that only one of the parents has the disease. So anytime that you see just a few individuals with the disease in a population, that probably tells you it is recessive, because if it's dominant, it's going to pop up a lot more frequently, because you have to be homozygous recessive to have a recessive disease. In other words, you have to have two of the same allele, and that's just going to be less likely. But that also means that anybody in the ancestral history of a person with the disease is going to be a carrier. Otherwise, there's no way that they could have even got it. So all of these people that have direct ties, are carriers for the disease. We know it's X-linked, so they have to be female. So all of these females in this pedigree are going to be carriers. So let's look at this. We have a child, so Chad down here has Hunter syndrome. But Bill and Sarah, neither of them have Hunter syndrome. So they want to know what the chances are of having any more children with the disease. So let's figure out what the gametes are for each parent. So mom, she must be the carrier because this is an X-linked disease. If dad had it, he would have the disease. But since he doesn't, we know that he's not a carrier because that's just not possible. So remember that these are sperm. So this is just, possible. These are eggs over here because they're from mom. So this is potential children. So all of these are their possible gametes, and anything within this box are going to be their potential offspring. So you can see Chad, they had Chad, he has Hunter syndrome. They only had a 1 in 4 chance, or a 25% chance of having a child with the disease. They have a 50% chance if they have a daughter of that daughter being a carrier. So, if you were to counsel this family, you would probably tell them that your risk is pretty high for maybe not conferring the disease to a child, but to keeping it within your family line in the future. So they might opt for not having children. So any man with the allele for Hunter syndrome in this family is going to express the disease. So a simple genetic test could tell you if you're going to get it, even if you don't, even if it's a disease that you don't display right away when you're born. Any woman that are ancestors of the men that have it are going to be carriers. So all of the female that are like the mom, grandma, great-grandma, great-great-grandma of Chad are going to have the allele. They're the ones that, even though it's like 4 generations later, still were carrying it, and they passed it on to Chad. And you can fill in, for the most part, a lot of these other boxes. There are going to be some unknowns because you could have a daughter who's a carrier that we saw from Bill and Sarah, but you can also have a normal daughter. So you can't figure out everybody, which is why genetic counseling gets difficult, because you can only predict the likelihood. You cannot predict exactly what is going to happen unless you test the actual DNA of the offspring in question. Okay. But Jessica and Steve, if they are trying to consider whether or not they should have kids, Jessica is still in this line where all of these women were carriers. We know that Helen and Carol were carriers. Jessica, there's a 50-50% chance that she is going to be a carrier. Steve, he is a guy, so since he doesn't have it, that means that he is not going to pass the allele on. But what is the likelihood, and what would you tell Steve and Jessica on whether or not they should have kids? So I want you to think about this one on your own and see what you think. And you can kind of go through the possible Punnett squares for them. Okay, so take some time to do that, and then I will give you the answers here. So on the left-hand side, if Jessica is not a carrier, that means that there is a 0% chance that they would have any offspring with Hunter syndrome or even a daughter that is a carrier. So if Jessica doesn't have the allele, then obviously they're good to go because both of them are completely healthy. But if Sarah has the Hunter syndrome allele, just one little H, she's a carrier, right? And we've done this same Punnett square a couple slides ago. There is a 25% chance that they would have a son with the disease, and there is a 25% chance that they would have a daughter who is a carrier. So maybe you would tell Jessica, you should probably get your genetic testing done on yourself to see if you are a carrier or not. And that would ultimately be the deciding factor for whether or not they should have kids. Now, thankfully, we live in a day and age where testing is possible. So you can test through amniocentesis. You can take some of the amniotic fluid in that sac surrounding the fetus. Obviously, that is a needle that has to go through mom, so that's not completely uninvasive. Can be quite painful, but you can check certain chemicals that would be like a marker for a disease just from that fluid. But you can also take off, you can gather fetal cells that are actually within that amniotic fluid as well. So you can directly test the DNA of the fetus by extracting some of the cells from the amniotic fluid. Chorionic villi sampling, that is going to be from the placenta. Remember that both mom and the baby contribute some actual tissue to the placenta. So if you take a portion of the fetal side of the placenta, that means that you're taking actual cells that developed from the fetus. So that's going to carry all the same DNA that will be in the body cells of the fetus. And those are rapidly dividing cells, which allows you to pretty easily do this testing. And it's a faster way to do it. You're not putting a needle in, but you still have to put like a suction tube up into the cervix and suction off a little bit of that chorionic villi, some of that tissue around there. So both of these are still going to pose a danger to the mom and the baby. So this isn't really recommended to just like everybody do this and then we'll make perfect babies by figuring out, like what's wrong and do like genetic testing on everyone, right? But this is a way that if you know you have a disease that runs in your family, you can get testing done to prepare yourself and also, any possible future that that fetus could have, you want to be able to be in the best position to provide for that child. Okay, that's all I have for you for this chapter. So go through the questions on your own. If you have any questions, let me know. Hopefully this was basically a review for you, but expect to see some form of like a Punnett square or a pedigree. Since we're on Canvas, I'm not gonna have you like fill in a pedigree, obviously, but there might be some questions where you look at a pedigree chart and you kind of make inferences from that. So just kind of be prepared to do that. Otherwise, have a great rest of your day and your week and your summer. And I hope you enjoyed this course. I think we all probably would have enjoyed it more in person, but I'm glad that we could all stay safe this way. And hopefully you are staying safe and healthy at home. So have a great rest of your week, guys.